Okay, let's dig into this. We're doing a deep dive today on advanced prostate cancer,
Yeah.
specifically mCRPC. Things are really moving quickly here.
They absolutely are. And we've got some fascinating updates. Our sources cover everything from boosting the immune system to, well, next-gen targeted drugs, and even totally new ways to design medicines.
Right. So our mission today, it's to really break down four key developments for you.
Hmm.
We've got two major clinical trial updates, things happening right now, and then two really cutting-edge preclinical platforms, things that show you where the future is heading.
So you, the listener, should get a really clear picture of the innovation pipeline.
Exactly, from current practice to what's next.
Okay, let's start with the clinical trials then, where the rubber meets the road. And the first one is, it's about making existing powerful treatments even better, right? Combining therapies.
That's the one. We're talking about combining radioligand therapy with cellular immunotherapy. Specifically, lutetium-Lu-177 vipivitide tetraxetan, which most people know as Pluvicto.
Okay, Pluvicto, got it. With Sipulecel-T. This is being looked at in a randomized phase 1 study run by City of Hope and the National Cancer Institute.
So Pluvicto, that's the radioligand, targets PSMA-positive cells with radiation. And Sipulecel-T, that's the immunotherapy that trains the patient's own immune cells against PAP, prostatic acid phosphatase.
Correct. It's an autologous therapy.
Seems like a smart pairing.
Yeah.
But what's the specific thinking? Why should the combination be better?
Well, the rationale is actually quite elegant. The Pluvicto, the radioligand part, it doesn't just, kill the cancer cells. It's thought to cause what's called immunogenic cell death.
Immunogenic, meaning it wakes up the immune system.
Precisely. When those targeted cells die from the radiation, they burst open and release these signals, danger signals, technically called DAMPs, like ATP and HMGB1.
Ah, okay. So the dying cells basically send out an SOS.
Sort of. More like they spill their contents in a way that really alerts the immune system. It fundamentally changes the local environment around the tumor, what we call the tumor microenvironment.
So Pluvicto clears the way, exposes the tumor's identifying flags, the antigen.
Exactly. It makes the tumor suddenly much more visible, and then Sipulecel-T comes in.
Right. To train the immune cells
Yeah.
to specifically go after one of those flags, PAP.
It takes the general alarm raised by Pluvicto and focuses it into a targeted attack. Very clever strategy.
Now, here's something interesting in the trial design I saw. They don't give them together at the same time. Sipulecel-T starts later, like week 8, after the first Pluvicto doses. Why wait?
Yeah, that's based on some smart preclinical work. The idea is you need the Pluvicto first to do that priming, release the antigens, get the environment ready.
Okay.
But Sipulecel-T introduces fresh, activated immune cells. You don't want to immediately blast those sensitive new cells with too much radiation from the Pluvicto.
Ah, so sequence it to protect the new immune soldiers while making sure the battlefield is prepped.
That's the thinking. It's about maximizing the synergy without causing unnecessary harm to the therapeutic cells themselves.
And the main goal of the study really hammers this home, doesn't it? It's not just about shrinking tumors.
No, the primary endpoint is actually the anti-PAP immunoglobulin G response rate. They are measuring how strongly the immune system ramps up against that specific target.
So they want hard proof the combination is actually activating the immune system as intended. That feels like a big deal.
It really underscores this move towards using immunotherapy strategically alongside things like targeted radiation.
Okay, let's shift gears. Subject 2, still in clinical trials, but a completely different kind of approach, tackling resistance head-on, the RIPTAC technology.
Yes, RIPTAC, specifically the drug HLD-0915. This is in a first-in-human phase 1/2 trial. It's for mCRPC patients who've pretty much run out of standard options.
Then what makes HLD-0915 so different?
It's what they call a first-in-class therapy. It uses this regulated induced proximity targeting chimera RIPTAC technology. And it's designed specifically to address, well, the main reason current treatments eventually fail.
Which is resistance, usually related to the androgen receptor AR.
Yep.
Cancer just finds ways around the drugs.
Exactly. Most standard drugs inhibit AR signaling, but the cancer adapts. RIPTAC tech is different. HLD-0915 targets both the androgen receptor AR and another protein called BRD4 simultaneously.
BRD4, what's its role?
Think of BRD4 as a kind of master regulator, a non-AR driver that cancer cells rely on, especially to survive and resist when AR signaling is blocked. It's a key escape route.
So hitting the main road, AR, and the main escape road, BRD4, at the same time with one molecule.
That's the core idea, trying to shut down progression and overcome that adaptive resistance that makes single-target drugs less effective over time.
And the early results, I saw the data presented at AACR-NCI-EORTC, look pretty impressive for such a tough-to-treat group.
Yeah, the results were definitely encouraging. They reported 59% of patients getting a PSA 50 response, so PSA cut in half, and 32% got a PSA 90 response, which is a deep response.
Wow. For patients with few options left, that's significant.
It is, and maybe even more striking, they reported that all patients who had measurable soft tissue disease showed at least a partial response on their first scan.
That's really something. Okay, so effective dual targeting, decent safety. No wonder it got FDA Fast Track designation.
Right. It really highlights this strategic shift, moving beyond just hitting one target to attacking the cancer on multiple fronts simultaneously.
So these clinical trials show some really smart thinking about combinations and hitting resistance pathways.
Absolutely. And, if we connect this back to the bigger picture, these clinical successes don't happen in a vacuum. They're built on some incredibly clever science happening earlier in the preclinical stage. The challenges seen in the clinic, like resistance, they really push researchers to invent entirely new tools.
Okay, so let's look at that next frontier then. Preclinical advances. Subject 3 tackles a notorious target, right? ERG.
Yes, ERG. This transcription factor is a major oncogenic driver found in almost half of all prostate cancers, often linked to the more aggressive types.
And it was considered, what, undruggable?
For a long time, yeah. Unreachable by medicinal chemistry was the phrase often used. The reason is, ERG just doesn't have the usual pockets or grooves on its surface that traditional small molecule drugs need to bind to. There was no obvious place for a drug to latch on and inhibit it.
So if you can't block it,
Yeah.
how do you stop it?
You get creative. The breakthrough here is a technology platform called Helicon Degraders.
Helicon Degraders. Okay, what do they do?
It's a really neat design. They develop these special peptide-based molecules using a stabilized helical structure. This structure lets the molecule bind directly and very specifically to ERG, even without a traditional pocket.
Okay, so it grabs onto the undruggable target, but then what?
Then comes the clever part. The Helicon molecule is designed to also grab onto another protein called an E3 ligase.
The E3 ligase, isn't that part of the cell's, garbage disposal system?
Exactly. It's the cellular machinery that tags unwanted proteins for destruction. So the Helicon acts like a bridge. One end holds onto ERG, the other end flags down the E3 ligase trash truck.
And the E3 ligase marks ERG for disposal.
Precisely. It tags ERG with ubiquitin, which signals the cell's proteasome, the disposal unit, to come and completely chew it up. Gone. Deleted.
So you're not just inhibiting ERG, you're actively deleting it from the cell.
Yeah.
It's a fundamental difference.
It is. It's aiming for complete elimination of the driver protein. And the results in animal models were pretty dramatic.
How dramatic?
They saw over 90% depletion of the ERG protein after just one dose. This led to strong tumor growth inhibition, and the effect lasted for at least a week.
Wow. And considering ERG is often linked to resistance against standard AR drugs.
Right. The potential is huge. Imagine combining an ERG degrader like this with an AR pathway inhibitor, maybe even an AR degrader. That could be a really powerful way to tackle resistance.
Okay, that makes sense. Eliminating tough targets. Now, our fourth subject, the precision platform, also seems to be about hitting harder and smarter, but through delivery.
Yes, precision focuses on precision delivery, but with a twist. It's described as a dual-payload peptide drug conjugate technology.
Dual-Payload, meaning one molecule carrying two different therapies.
That's right. And it addresses two key challenges: getting the drugs only to the tumor and dealing with the fact that not all cancer cells within a tumor are identical. That's heterogeneity.
How does it target the tumor specifically?
It uses a protein called FAP, fibroblast activation protein, as its target. FAP is found in abundance, specifically in the tumor microenvironment of many cancers, including advanced CRPC, but not much in healthy tissue.
So FAP acts like a postcode for the tumor.
A very specific postcode, exactly. The precision molecule is designed to find that FAP postcode. Once it binds to FAP, an enzyme there cleaves the molecule, releasing the two therapeutic payloads right inside the tumor microenvironment.
Okay, targeted delivery. And what kinds of dual payloads are they packing?
They can mix and match. For instance, they've combined microtubule inhibition using a drug component called MMAE with topoisomerase 1 inhibition using exatecan.
Two different ways to kill cancer cells.
Right. Or another powerful combo pairs topoisomerase 1 inhibition with inhibitors of the DNA damage response, DDR, things like PARP inhibitors or ATR inhibitors. Many advanced prostate cancers have defects in DNA repair, so hitting that pathway along with topo 1 can create synthetic lethality.
And the benefit of delivering two at once.
Synergy. The preclinical data showed that this dual payload approach could kill 4 to 5 times more tumor cells compared to using the same drugs individually.
4 to 5 times. That's a big jump.
It is. And there's another critical aspect. When the payloads are released by FAP cleavage, they don't just kill the FAP-positive cells they initially targeted, they diffuse outwards and kill nearby cancer cells too, even if those neighbors are FAP-negative.
Ah, the bystander effect. Tackling that tumor heterogeneity problem we mentioned.
Exactly. It helps ensure you're hitting more of the diverse cell populations within the tumor, not just the ones displaying the initial target.
Okay, let's try to wrap this all together.
Mm-hmm.
We saw clinical trials focused on smart sequencing of therapies like Pluvicto, then Sipulecel-T for immune priming.
Right. Then we saw a single drug, HLD-0915, using RIPTAC, achieving dual-axis targeting of AR and BRD4 to combat resistance.
Then in preclinical, we saw Helicon degraders designed to eliminate previously unreachable targets like ERG.
And the precision platform using FAP targeting for dual payload delivery to maximize synergy and overcome heterogeneity.
The common thread seems pretty clear.
It really does. It's all about mechanical complementarity. The focus isn't just finding one new drug anymore. It's about designing combinations, whether sequenced or simultaneous or within a single molecule, that work together in ways that overwhelm the cancer's ability to adapt.
Combinations are becoming the central strategy.
It seems that way. Designing for synergy is key.
So the final thought for you, our listener. We've seen all these advanced strategies, combination, dual targeting, dual payloads, really taking aim at resistance in late-stage mCRPC. It makes you wonder, doesn't it?
It does. If overcoming resistance requires these complex, multi-pronged attacks in advanced disease, what does that imply about the future? Will we need to start thinking about using these kinds of combination or complementary strategies much earlier, even when the cancer first appears, to prevent resistance from ever taking hold?
Something to definitely think about. Thank you for joining us for this deep dive into the source material today.
Stay strong and fight on.